Okay, let's continue. Let's dive into the question, what other sources of variation are there? Well, browsing through literature, at some point I came across this paper which gave a very nice overview of all kinds of sources of variation. I really like this one. It saves me tons of time thinking for myself. But I think this is what we're looking at. Right, overall variation, we can, well, decompose it into different contributions of a different nature. So it's at the left, we have the biological variation. Here we might have what we might call the pre-analytical variation, analytical variation, and post-analytical variation. And the thing is that the variation in which we have an interest, the thing that we want to have is the biological variation. That's what we're aiming for. This is what we need in our experiments. If you do not observe this biological variation, the thing I call biological variation, your experiment did not deliver. You have basically measured all the time the same thing. It's not working out. Well, if you go a little bit deeper into biological variation, what are different factors that contribute to biological variation? Well, intrinsic, extrinsic factors within person variation. Well, for person, you can also read any experimental unit you have. Concentration, size, volume, weight, all things that make up your sampling. The variation you don't want is in red. This is a variation that you feel like you could do without because it comes on top of your variation in which you have an interest. Yeah. If the one in red is not really existing, you would have high data quality. But the more variation that you introduce down the road, the more, well, you could call it experimental bias you will introduce into your experiment. So maybe we should go a little bit deeper into this type of variation. So if we look at pre-analytical variation, it's basically variation that you have before you do your sample measurement, or everything that contributes to before you do your actual measurement. So this is about sample collection. This is about handling, storage, stability, extraction. This is about all these things. And the message is that tiny changes lead to additional variation. So things that you, well, maybe unavoidably change over here, it will lead to tiny additions of the variations that you may, well, you don't want to have it over there. The other one, analytical variation, well, it's related to the sample itself, the measurement, the instrument, method parameters play a huge role in this. So column, mobile phase, system stability, batch effect is a big one. We can't measure everything in one run, so we need to do multiple runs, how to deal with that. Matrix effects, different labs, different operators, different somethings. All these things, sample degradation, all these things add a layer of variation to the variation in which you have the specific interest. And how can you counteract these things? Well, working with standardized conditions, standardized protocols, doing every time, all the time, doing the same thing. But, well, I'm not an expert in doing all these things. But my guess is it's very, very difficult probably to do it exactly the time, all the time. So you will have to deal with the variation. So what can we do? Well, the message is we will get unwanted variation. It's too difficult to get rid of it. But what can we do? Well, sometimes we can recognize the source of variation unwanted variation that we might have, and then we can actually control the unwanted added variation, right? What we can do is recognize the structure that we have in the experiments. And a very simple example of this concept is doing experiments in blocks, or in fields. Maybe that's a natural word. Do it in fields. In agricultural experiments, then I just took two random pictures I stole in Google. And this is a very typical setup. You have a certain field, and you are trying out different varieties or different regimes or different strategies or whatever. And usually it's about yield. We would like to have as many potatoes as possible for some reason. And but sometimes, yeah, we cannot do all our experiments in one particular field. And then you have to incorporate multiple fields. Yeah. Well, fields are never alike. Yeah. Going from one field to one field, there might be differences between the fields. So that's a source of unwanted variation. There is what we might call a field effect. Now, there is a way to deal with that, because once you recognize that there's a source of unwanted variation, being the field, what we could do is say, well, you know, what we're going to do is we'll introduce all our treatments in both fields, and to the model we'll add a factor. Yeah, I'll come to modeling at some point. Then we'll add a factor that will describe the field effect. Yeah. So instead of ignoring the effect of field, yeah, we are actually going to incorporate it in a model. We know it's there, and we try to get rid of the variation in a model perspective. What would be, by the way, the worst thing? Can somebody help me if you do this experiment in two fields? Yeah, what would be the worst way to do this experiment? Suppose you have an interest in two varieties, A and B. You want to know which one gives you the most potatoes?
It's one variety.
Yeah, that would be strong in the experiment, right? It's, you have confounding between your field effect and your variety. But now difficult question maybe, or a more difficult question. How would you do your experiment? Say you have two fields, two varieties. How would you do it in a correct way? Difficult question. Maybe somebody can help you.
If we want both varieties.
Exactly. That's what you need to do, right? In every field, you need to put both varieties, and then you can actually do a block design. What you're actually doing is you're comparing the varieties within the field, thereby ignoring the field effect. That's the idea. Well, that's what we call a block factor. Field in this example is what we call a block factor. And examples of block factors is, for instance, proximity in space. I guess that's the field example I use. But it can also be proximity in time, right? Stuff that you measure on the same day or in the same week, that is more equal than stuff that you measure in the next week. So that would also be a block factor. So say you have, well, let's pretend you have 5 treatments, yeah, and you want to do, want to measure 5 treatments. Well, what you could do is do one treatment in 1 week, and the second treatment in the second week, and the third treatment in the third week, and so on. Yeah, well, that's confounding. But what you can also do is recognize that week will be a block factor and do every treatment in every week and just repeat it for as many weeks as you need replicates. Right? And that will be something you could do. But it can also be about similar physical characteristics. Yeah. So suppose you are doing research on, and you have to come up with examples. Suppose you are doing research on yield. Many of the stuff it's about yield, but about peanut plants. And peanut plants, yeah, in the beginning of an experiment, they vary in size. They're not all of equal size. They thought to do that. But you know that peanut plants that are bigger, they will get more peanuts. Yeah, and then you apply treatment, say a fertilizer. But what you could do is actually recognize that bigger plants at the beginning of the experiment will give you a bigger yield anyway. So if you record the height of the plant at the beginning of the experiment, put that in your model as covariate, you actually filter out the unwanted variation because of the different sizes of the plants at the beginning of your experiment, right? That's what people call analysis of covariance. You just add a covariate to the model, and that would work like a charm. Yeah, this is also one of the placeholders for me to say that when you do an experiment, try to record as much of the things you can, because later when you go to the statistical modeling, you might know at the beginning what you would like to include as covariate in your model. Yeah, I'm not sure if people automatically record plant height or such a thing, or weight or whatever, but it's probably easy to do, and you might add it to your model. If it's significant in the model, it filters out variation you don't want there. And if it's not significant, well, you have learned something that differences in plant height don't really matter for what you were studying. Right? So that's also something you do. Yeah. So, well, if you do add a block factor, and block factors can be fields or days or length of plants. It can also be machines and operators. Those can also be block factors. What you are doing basically is a randomized complete block design. Yeah, it's called randomized because within each block you still randomize all your treatments. Yeah, you're not going to do treatment 1, 2, 3, 4, 5 all the time. 1, 2, 3, 4, 5. That's confounding. Yeah. So it's randomized. It's called complete block because you're supposed to do every treatment once or twice or 3 times, but you're supposed to do every treatment in every block. Yeah, and it's a design. Yeah, I guess the word complete also gives away that there must be something like an incomplete block design, a randomized incomplete block design, which is true. And that would be the situation where you cannot do all the treatments in every block because the blocks are not big enough, technically, literally probably. And then you do incomplete block design, but also remember that there are clever experimental designs that tell you do these experiments in this block, these experiments in this block, and on and on. And then the statistics in a clever way will do all the comparisons for you with the maximum power, right? Randomized incomplete block designs. Okay, let's do a challenge. Yeah, let's get our brains. This is a snippet of a story about potatoes. We have 4 types of fungicides. Yeah, we want to test them on the yield of potatoes. And we also have a control. There's no fungicide there. And a single field is not big enough, so we're going to use 4 fields. Yeah. So I have a couple of questions. How many factors are in this experiment? I hear somebody say 6. 1 factor. Type of fungicide. Yeah, that's definitely a factor.
And then it has 5 levels.
And it has 5 levels, but I was still looking for another factor actually. Field is also a factor, right? That's the funny thing when you do block experiments. Yeah, you have an interest in your factor fungicide, which obviously has 5 levels. Yeah, for fungicides and the control, it's also a level. So there are 5 levels there. But yeah, since you do it in different fields and you incorporate field in your experiment and later on in your model, it means you have 2 factors now. Yeah, because you don't want it, but you got it. Yeah. Another question: what are the experimental units? And experimental units is like statistical lingo for what we probably call samples. Do you know, by the way, that the word sample also has a different meaning for statisticians? They call a collection of observation also a sample, which is sometimes, if you're not aware of that, it really breaks down communication. But so if I talk with other statisticians, I always say sample in the meaning of a collection of observations. But if I talk with people doing experiments, I just call a sample, and then never mention the word sample in the meaning of a collection of observations. But be aware of that when you read some literature. But what are the experimental units? Let me help you. It might be difficult questions. You're not used to these type of questions, but experimental units are what is actually receiving the treatment. So you said potatoes? Yeah. Any other choices? So what is actually receiving the treatments in this study? This snippet of a study? The fields. Yeah, the fields are receiving the fungicides, right? So it doesn't matter for the experiment. Everybody does it correct when you're doing these things, but yeah, the experimental units are the fields. So if you have in a field, say 10 plants, then you don't have, say, 10, you don't observe 10 numbers. Yeah, you observe one number coming from the field, so you're probably going to take all the potatoes from the ground and just weigh them. And that will give you per field 1 number. Yeah. Yeah, and then the potatoes, I guess, would be the measurement units, but that's becoming a bit cloudy. Now, the challenge continues because I had a master student once. Now, let's make it a bachelor student at some point, and the bachelor student asked, well, please propose this is not yet. Please propose an experimental design. And then the student came up with this. It's block design. Let me talk you through it. Yeah, we have 4 blocks, 1, 2, 3, 4. We have 5 plots because it's a randomized complete block design. All the treatments must be incorporated somewhere. So 1, 2, 3, 4 of variety 1, 4 of 2, 4 of 3, and then the controls, and then we use the last remaining options to do variety 4. I think it's very doable, right? If you are in the field, yeah, you are in one block, you only need to bring variety one and a bit of 4, and then you put it in the ground. I'm getting the feeling I'm really overdriving this point, but no, I have seen so many funny stuff that I just keep adding in these slides, because the big message is when you are doing your experimental design or your measurement design, and you see any irregularity in this design, in the numbers that you write down, in the order of things, there is a problem. I can almost promise you that there is a very specialized design excluded. There is a problem, yeah, because this is very irregular, right? 1, 2, 3 control, and there's a column of 4. This is very irregular, but this means that you're introducing confounding, right? Variety 1, 2, 3, and the control. They're never seen outside of the specific blocks they're in, and that's a problem, right? That's not what you want. Now, if you want to improve the design, right, this is what we need to do: a randomized complete block design. So in every block, we have every treatment, and we randomly assign the treatments to the plots in the field. And then it works out nicely. And the big question for you would be, of course, how do I translate these ideas, these concepts, into your specific research? Because I'm guessing nobody is in the audience is doing yield on potatoes and fungicides. Yeah, but shows an easy story so everybody could relate to it. Yeah, but that will be the challenge. Yeah. Now there's a pitfall working with block factors. Yeah, the pitfall is that your block factors are confounded with your factors of interest. That's a bit of a summary. I said it a couple of times. The answer to that is randomized, but let me tell you about this specific case story I came across on the internet, literature. What people did at some point, and it's a bit like a phrenology, right? Where they look at the lumps of the skull and then attribute all kinds of properties to these persons. But what these people claimed is that they built like a machine learning or AI application. They were able to, based on photographs of persons, they were able to predict if these people were engaged in criminal activity or not. It's a bit of a scary topic, I'm sure, but nevertheless, you can find it still there. And the paper actually has like 6 photographs in there, and of 6 persons. And yeah, I thought for today, let's copy paste them. But the paper doesn't even do the black bars across the eyes, so I felt uncomfortable not doing that. So the bars are my own. But then, well, they were able to, so these are like 3 samples from criminal ID photo set. Yeah, these criminal photos, they came from, I'm not sure what you call it in English, but the photographs that were taken at police stations, etc. And these 3 photos are examples of non-criminal photos. And if you read the paper, they were taken from professional photographs that people may or may not have on their company websites. Yeah? And they built this machine learning application, and they were perfectly able to predict criminal behavior versus non-criminal behavior. And they wrote a paper about it, and they said, yes, we can do this. Excellent application. And well, let's become famous researchers with this topic. Yeah. Can anybody spot the problem?
Non-criminal is always smiling.
Yes. I think you're onto something. I think this is the basis of the problem, right? Yeah. No, this is definitely it, right? This is definitely it. Because what did they do? They went to photos taken at police stations after people were arrested, and they had their mugshots taken, and they compared these photographs to people that have done the photos on the internet. Yes, so everybody can see who they are if they have an appointment or whatever, you know. So that's confounding, right? It's if you want to do this properly, you should get everybody a mugshot at the police station, or you should get everybody's professional photo from the company website, or you should not do this type of research, maybe. I'm not sure if that's possible, but you know, this is a very strong example that is, yeah, even nowadays people don't recognize the confounding that they introduce by the data collection. Yeah, so it's very important. Yeah. Now let's talk a little bit about modeling. If we do an experiment, before the tea break, I said that usually when you do an experiment, you keep everything constant and you only change the things in which you have an interest. So you keep temperature, moisture, whatever, keep it constant, and you might actually change, I don't know, maybe your light conditions, no light, high light, medium light, whatever, and then you start to analyze your model. Well, the ideas that you have about your experiment, they are actually reflected back into your statistical model. Yeah, that's the idea. So in the example, of this fungicide, suppose that we try to keep everything constant, then the only thing that you're going to change is your fungicide and the block factor. You have two factors, and these two block factors, they are reflected back into your model. Now, this is assuming that we really were able to keep everything constant. It's probably also not the case yet. So how would you read this particular model? It's an additive model. So the response for a certain fungicide in a certain block, it's equal to all kinds of additive effects. For instance, there is some sort of a mean, a sort of reference value, and on top of that, you add the effect of a fungicide. And I say adding, but it could also mean subtracting if the fungicide is not really that helpful. And then on top of that, you add a block effect, because maybe some blocks are in more fertile soil than other blocks. And then, of course, we have the errors that we make, stuff that's not contained in this. But you see that this model actually contains all the information that we have about the experiment, because we kept everything constant, and only the things we changed, they are back in the model. That's the idea. Now, if you want to do some statistical analysis, say you want to know if there are any differences between the fungicides, what you do is simple hypothesis testing, for instance, on the parameter estimates coming from this model that tell you if there are any differences between the different fungicides. Now, there is this term, I quickly glanced over it. It's called the residual term, and it's becoming a bit more statistically, but promise that won't last more than 5 minutes. These residuals, they contain all the variation that you could not explain, with the factors of your interest. So if you were able to keep everything constant, change only fungicide, and you have a block factor, then your residuals are probably okay-ish. There will be a certain amount of them, but it's fine. But say that are all other kinds of variations that you don't want to have, the pre-analytical variations, sample variations, the post-analytical variations, those type of things. All these variations, they will go into the residuals. And why is this a bad thing? Well, because the residuals, the errors, those are the actual within errors, they will be used to compare the between distances, the between variation. So if your residuals become bigger and bigger and bigger because of all this variation, then from a model perspective, at some point you don't see any significant difference anymore. And at some point, that's what we want. Well, these residuals also need to meet certain assumptions, but I'm going to ignore that. That would make it in the statistics course. It's probably not making you too happy, I'm assuming. So no, I want to talk about hypothesis testing, because hypothesis testing is basically the thing that connects. Yeah, there's a question.
For designing an experiment, we should always consider the linear model.
No, not really. Sometimes the system is non-linear, and you might see a linear and then certain decay or anything. You might want to make a model, but most models are linear because it's already difficult enough to work with linear data.
Because if you are keeping all the other parameters constant, if you are just focusing on one parameter, but if you consider the environmental samples, then it's very difficult to just change one parameter. Because in environmental sample, many things heterogeneity always comes in between. Because if you take sample from here, you take sample.
If you take one thing, something else changes with it. I agree.
So how to deal with that and then design this experiment? Like if you take an agricultural field, and if you take soil from here and soil from here, they both are different. Like in case of microorganisms, not like irrespective of.
Well, what you could do is observe many of the things that you are out of your control because they change with what you were doing. Yeah. And you could maybe add that to your knowledge covariate. Yeah, it's a variable that you add to the model to at least filter out some of the unwanted variation. But I feel that we're going into the operational science now. And because we cannot control all the variation anymore, then it's really important to be careful not to filter out variation that's related to your response, because that can also happen. But I think what needs to be done is that you start recording all the variation that you have. If you know that one thing change over here leads to change in all the other things, I would report them all and put that in the model.
But if there are outliers, like if you take bunch of different samples and if you do any kind of experiment and then you see outliers in few samples, and how can you say if that an outlier for an experiment or outlier there is something science and the outlier is coming?
Outliers are always a difficult topic, right? Because there are tests that you can do to say specifically this is an outlier, but I find it very difficult to do. The best way to detect an outlier is to go back to your experiment, to go back to your notes and to realize what I did. I made an error, or I took a measurement, a reading, and then somebody opened the door, slammed the door, and at the second moment I was injecting something, and I was got a bit of a jump scare, and this funny stuff happens. And that's like an observation that went wrong, and it's safe to remove those from your data. But without a really good reason removing data points, that is very hard. I'm not much in favor of it because I realize all the time how easy it is to get favorable results just by removing a data point, changing a number in a data set. And I find it super important to be super transparent in the highest integrity statistics because they have so much power. Because somebody is going to check your work at that level.
That's the thing, because if you're doing any kind of control laboratory experiment with controller features, if you're not following this linear model, like one thing constant and the other things constant and one thing changing. But if you take considering environmental samples, then you cannot repeat the same experiment again. So that's the thing you can see. That's how to make an hypothesis.
We will talk about that in the tea break or in that.
Yeah.
Yes, I have some ideas too, because you also talked about the algae and the glaciers, right? Yeah, sure. Let's do that. Yeah. So this hypothesis testing is touching back upon what you want to do. Yeah, it touches back to your research question, and what it is doing, it is assessing the variation between the treatments, and it is comparing it to the variation that you have in your biological replicates. Yeah, that's what doing. So what you could look at, the hypothesis test is, for instance, a bit like a signal to noise ratio. Yeah, the signal would be, say, the difference between the groups. Yeah, you have two treatments, so there's a difference between the two groups, and then you are going to compare it to the variability of the groups themselves. So how much variation is there in a group compared to how much difference is there between the groups? And if you would like some formulas with that, you could write X bar treatment, and that would be the mean of the treated group minus X bar control, the mean of the not control group. And then you divide that by the standard deviation of that particular difference. And if you do that, you are doing a t-test, because this is a definition of the two-sample t-test. Yeah, I put it over here for completeness sake, but that's the basis. Yeah, it is just a signal to noise. What you do all the time is, what is the difference that I found, and what is the variability in the result that I have if I were to do it again. Yeah, that's what you compare. Now, you can also look at it from a very pragmatic point of view, because the difference between two treatments, well, if it's zero, it's not significant. You don't need statistics for that, hard zero. If the difference is very small numerical value, you also don't need statistics. It's not going to be significant. If the difference is small, it's probably not significant. If it's a bit larger, then it's maybe significant. If the difference is pretty large, yes, it's quite likely it will be significant. And the difference is huge. Yes, this is without a doubt significant. So without doing fancy statistics, you can also use this lookup table. Yeah. No, not really. You need to finish it properly. Well, unless I can get this idea published, and then you can cite it, and then it's fine, right? But the thing is that most research seems to be in categories 2 and 3 and 4. So we're always looking at a very small, and sometimes a bit larger differences, right? And those are the difficult things to find, because we all know when something hasn't worked well. It's easy to spot. The big differences, don't think they exist anymore. I think all the low-hanging fruit is gone already. Maybe I'm just pessimistic, not sure. So we're really in this particular area. Now, this t-test idea of the signal-to-noise ratio, I even showed you the formulas, is a very general principle. It's something you encounter in comparing one sample with a predefined number, in comparing two samples. If you do paired data before and after measurement, say you have one plant, you take a leaf before the treatment and you take a leaf after the treatment. Well, I need to think careful about such a design, but that's in principle a paired measurement. But you can even test correlation coefficients, right? I'm not sure if you're aware of that. Let's do a quick test. Who is aware that you can test if the estimate for a correlation coefficient that you find, that you can actually test it if it's significantly different from 0? Who knew that? Okay, because you will always get a numerical estimate, right? Computers will always spit out numbers. That's no problem. So you might find that, oh, my two variables correlate with 0.8. For most applications, that's very high, makes you very happy because you get a story to tell. But the 0.8 might, statistically speaking, not different from 0 or true 0 because of the variability that you have in your data. Just that you get a numerical estimate might make it not significantly different from 0. Yeah, so you can do a t-test on the correlation coefficient, see if it's truly different from 0. That's what you need to do. If you do a linear model, you can test your regression coefficients. It will tell if there is truly a relationship between what you want to predict, what you have as your explanatory variables. So this t-test should be useful. Yeah. So the statistical testing in a nutshell, what do you do? Well, it starts out with the null and alternative hypothesis. Yeah, the alternative hypothesis is the research hypothesis. That's the thing you want to prove. You want to prove, yes, there is a difference in this and this compound between a control and a treated group. Yeah, that's your research hypothesis. Otherwise, you wouldn't be doing it. If you don't truly feel that there are differences, then you don't want to do the research, I guess. And then there's the null hypothesis. That's basically the opposite. There is no difference. Yeah. Well, what you need is a p-value and a certain confidence level. And I'm skipping a few steps, but that's fine. Yeah. And the p-value, oops, that will be the wrong button this time. Yeah. The p-value is the probability of obtaining your data if the null hypothesis would be true. So if the null hypothesis is really true, there is no difference between the two groups. But if you have your data, then the p-value is the number that tells you, well, the probability that you have your numbers and the null hypothesis being true, that probability is 10 to the power minus 20. That's so rare, it's not going to happen. Well, we can do either one of two things. We can say, well, the data is wrong because the null hypothesis must be true. Or we can say, well, the data, that's the physical realization of the world around us. But the null hypothesis is a mental concept. So that one must then not be true. That's the last thing is what we do. Yeah, we just disregard the null hypothesis and we say, well, we have proven the alternative hypothesis. This compound is significantly different in these two groups. Yeah. Involved with this decision rule is a threshold. That's the threshold of reasonable doubt. This is the 0.05 that you keep reading about. Yeah. What we say, if there is a less than 5% chance of us observing the data, given the null hypothesis being true, if that probability is less than 5%, we say that's too rare, right? That's too rare. We're not going to do that. Yeah. Well, if you think about it, when you do hypothesis testing, you can make errors in there. You can make several errors because you can do a false positive. You think there is a difference, but in reality, there is no difference. And in my table, that would be over here. Yeah? So the conclusion of your test says we need to reject the null hypothesis. But in reality, which we cannot conceptually know, I guess, but in reality, the null hypothesis is true. Well, we make an error. Yeah, we make an error, and this is what we call a type 1 error. It's a false positive. And you know, false positives are really annoying because false positives, they let you believe that you're onto something, and you may or may not go into follow-up research and spend money and time on it, and then later on it's gone, right? It's super annoying. Yeah. Now you may wonder, what's the probability that we do a false positive? Well, it's in the decision rule. So as soon as the probability drops below 5%, 0.05, we are going to reject the null hypothesis. But occasionally, we come across a result that had this lower probability than 5%, but the null hypothesis is still true. Conceptually, that can happen. Yeah. So this means that in 5% of all the research, we will have a false positive. It's super annoying. 5% means 1 in 20. So if you do 20 t-tests, then you're bound to have one false positive. Yeah. And I think that this is one of the reasons why we see so many results in literature that are not reproducible. Yeah, I mentioned that maybe samples are not comparable, so there it's difficult to get the same results. But I also think that many of the results are actually false positives that have been reported into the literature. Yeah, because maybe no follow-up research was done or whatever. Yeah. So that's 5%, super annoying. Yeah. Now there's another error that you can make. There is an error that is the false negative. Yeah, you think there is no difference between the two groups for a particular compound, but there was, but your experiment failed to pick it up. Yeah, this is a false negative. So we are over here. The conclusion of the hypothesis test is do not reject the null hypothesis, but in the real population, the alternative hypothesis was true. Yeah, this one is also annoying, but at least you don't know it, right? You don't know what you don't know, so you're not spending, sobbing about what you were not able to detect. Yeah? But there's also a probability attached to this error, the false negative. People refer to this one, by the way, as a type 2 error. And there is a probability attached to a type 2 error, and that will be beta. Beta is the symbol for the type 2 error. And what we now do usually, no, wait a minute, I'll save that. Yeah. Well, okay, let's go down a different road. If we are doing hypothesis tests, and remember hypothesis tests are univariate, you do them one metabolite at a time. For typical data set, it means you're doing hundreds of t-tests, right? And if there's a 5% chance of getting a false positive, if you do it 20 times, then you're bound to have many false positives in your list of significantly different metabolites, which is super annoying. Yeah. As an illustration, I try to illustrate how severe the problem might be. I try to following a graph. The probability of making an error, the type 1 error, is alpha by design. Yeah, that's just by design. So the probability of not making an error, that will be 1 minus alpha. And 1 minus alpha is a quantity you may have heard of. It's what we call the confidence level. Yeah, it's the 95% usually. And the probability of not making an error in M tests, well, if these tests are all independent, and in metabolomics, that's not really the case because there's a huge correlation structure between them. So I'm already cutting huge corners here. But the probability of not making an error in M tests is 1 minus alpha to the power of M, because independent probabilities, you can multiply them. And then the probability of making at least one error in M test is 1 minus, between brackets, 1 minus alpha to the power of M. Now, this is something you can actually plot, and that's what I did over here. So here is N, the number of hypothesis tests that you are doing, and I just plotted from 0 to 100 because it became pointless after 100. And here is the probability of at least one false positive. And if you do only a handful of hypothesis tests, well, you are at 20%, 40%, that may not all be too bad. But if you do 100, the probability of getting a false positive is 1. It's guaranteed. You will have them. Yeah. And from metabolites, you will have more than 100 metabolites routinely. So you will have, well, a probability of 1 to have at least one false positive. But that's the catch. This is about one false positive, right? There's also a probability of getting 2 false positives and 3 false positives, which is super high. Yeah. So the bottom message and the bottom line will be, if you do hypothesis tests and you do them many times after each other, you will get many false positives. And it's a problem if you're going to use these false positives in, say, some follow-up analysis or research, because you might be investigating false positives. That's a bit unfortunate. Now, there is a way to reduce the influence of false positive. It's a very active field of research. I'm not involved in it, but occasionally something seeps through. What you want to do is reduce the number of false positives. Conceptually, you can never get rid of false positives, but you can at least reduce the influence of false positives as much as possible. And there are different ways that you can reduce the number of false positives. There is, for instance, a per comparison error rate. Yeah, so it means that it means that you're going to adjust the decision rule. You're going to adjust alpha. You're going to lower alpha even further to make it super difficult for a result to be called significantly different. Now, if you work with alpha 0.05, there's a 5% chance. But if you do alpha 0.001, yeah, there's a 1 in 100 chance that you get a false positive. So that already helps. Yeah. One of the methods that's doing this is called Bonferroni. I'm sure most of you are aware of it. It's a very simple thing. It's where you take your alpha and you divide it by the number of hypotheses that you are doing. Yeah. Now, it sounds all great, but it's not. Yeah. There are two big drawbacks attached to this. Yeah. I'm not a big fan of Bonferroni unless a false positive will really, really have detrimental effects. Because you know why? There are two reasons. If you work with very high dimensional data, say you're doing microarrays and you have 20,000 observations and you're going to do 20,000 hypothesis tests, you're dividing alpha by 20,000, your alpha becomes so low, nothing is ever significant anymore, except for the gene that is doing the black and white thing, which is already known from literature. So Bonferroni doesn't work in those settings. But also, what I don't like about it is that if a hypothesis test becomes significant or not depends on the number of other hypothesis tests you're doing. So it's more conceptually, right? If you were to do an analysis in a targeted way, you only measured one metabolite, that could be, then you don't adjust alpha 0.05. But if you measure that same metabolite in the presence of 10,000 other ones, it's not going to be significant anymore. And that's, I find, the conceptual problem with it. And so we'll probably be a little bit careful with it, yeah. But otherwise, it's fine if you know the drawbacks. No, I have much more faith in these methods, the family-wise error rate and the false discovery rate, because the family-wise error rate is basically the probability of making one or more false discovery or type 1 errors when you are doing multiple hypothesis tests. So it's lumping, it's setting some sort of maximum to the amount of false positives that you expect. And this is very comparable to the false discovery rate. Now, if you have a false discovery rate of 5%, it means that among all the features you call significant, at most 5% will be false positives, right? And that's something different than having 5% false positives of all the comparisons that you were doing, right? I hope you see there's a lower number in there. Yeah, so that's the idea. Yeah, so these techniques, keep them in mind when you are doing many of these univariate things. Now, if you are doing exercises, experiments that have more than two groups, very common, yeah. T-tests don't really work that well anymore. Yeah, what you want to do is go to ANOVA models, analysis of variance models, where you incorporate factors that have more than two levels, and then, well, you can go into F-tests and T-tests and so on. Yeah. So in this factorial ANOVA, what do? Well, we test for differences between treatments, and what we do is we decompose it in variance components and what we can do are F-tests. Yeah, so they're well connected to T-test in a sense, but they're F-tests, and the F-test is nothing more than the ratio between two variances. So you are really looking at the variance of the between variation compared to the within variation. But even this is like a signal to noise ratio, because the variance between your treatments, that's the signal. You want that variance to be as large as possible. But the variance inside the treatment, that's the variation you have in a group, you want that to be as small as possible. Yeah, and this particular ratio will tell us yeah, if it's going to happen or not. Yeah, that's the idea. Now let's go to power and finally get to the concept of power, because there are things you can do to increase the power of your experiments. That is something that might be of your interest. And you know what I try to sketch over here? I try to sketch the distribution of a certain metabolite. It's the distribution of the difference between a metabolite for two conditions. And in the null hypothesis, it's over here. Yeah, it's centered around 0. Yeah, and also under the alternative hypothesis, where we say, well, there is a difference. Yeah. And what I did, I color-coded yellow and green. I color-coded alpha and beta. And alpha, remember, is associated with a type 1 error, and beta is associated with a type 2 error, the false negative. Now, what happens? Suppose that the null hypothesis is really, really true, yeah, but you, for some reason, reject the null hypothesis. That would be a false positive. It would be because you find in your experiment a difference that is in this right tail, in the yellow area, right? And this yellow area, that has a probability of 0.05, so that will be your probability for a false positive. Yeah. Now let's reverse the situation. Suppose that the null distribution isn't true, but the alternative hypothesis is true in reality. Yeah. So what we are doing is we are sampling from this alternative distribution, but say that for some reason we fail to see that there's a significant difference. Well, from this graph, you can see that would happen if you get an outcome of your estimate, of your difference, if it is to the left of this dashed orange line, right? So for this whole green patch, you would not be able to reject the null hypothesis. Well, you would like to do that. Yeah, that's what we call that is the beta. That is the probability of a type 2 error. Now, what can we do? Well, first of all, let me point out that the type 1 and the type 2 error are like these communicating barrels with water in it. If you push on one, it goes up to the other end. Yeah, if you want to have fewer false positives, right? If you want to have fewer false positives, what you're going to do is you're going to decrease alpha. So alpha, this line is moving to the right, meaning that your green area becomes bigger. So if you make it more difficult to get false positives, which is a good thing, it at the same moment means that you have a higher probability of false negatives. So there's no free lunch. You can't fix it. Yeah, you either have false positives or you have false negatives. Yeah, that's a bit of a problem. But is there now nothing that we can do? Yes, there are a couple of things that we can do. If you specify, say, the effect size, if you say I'm interested in bigger effects, so these curves lie further away from each other under the null and alternative hypothesis, you can see that the area in which they overlap becomes smaller, and then the risk of false positives, false negatives becomes a lot smaller. So if you say I'm not going to do these tests for very small differences, because false positives, negatives might happen, that could be a start. Yeah. If you were able to decrease the variation, that would be great. I think I've been talking about variation and source of variation for an hour, maybe in total. If you lower the variation, what it does, it has the effect of making these distributions smaller, right? So they become smaller and smaller and smaller. And if they become smaller, the area of overlap, that would be this triangle, compared to the bigger one, is smaller. And that would also be a great thing. But I also know that it's not always possible to decrease variation. It's super difficult, and it is usually what it is. Yeah. But the biggest thing is over here in bold. Yeah, you need to increase your sample size. With bigger sample sizes, you achieve the same effect that your distributions become more narrow. Yeah, and yeah, that means for the same reasons you will have less overlap, meaning smaller probabilities for false negatives and false positives. Yeah. Well, when you design experiments with power, typical numbers that people like to do is the probability of a false positive 0.05 and the probability for a false negative 0.10, 0.15, 0.20. Those are typical numbers. So it means that you accept a 5% chance for a false positive and say a 20% chance of a false negative. Yeah, and if you hold these numbers, and there is software, it's called G-Power, it's free on the internet, looks like a charm. If you type in these numbers, usually a sample size comes out of it, but you can also do the reverse thing. If I can only do 30 samples, and these are my properties, like the variation I have, what would be my observed power? Because power is defined as 1 minus beta. Yeah, and that could tell you if it's worthwhile to do the experiment or not. Yeah. Okay, well, brings me to the last slide. And I feel experimental design, let's do a wrap-up, some sort of conclusion forming. And I feel that experimental design is an important part of research. Yeah, I hope that all the items I crossed off the list helped you convince to do that. Yeah. Take care of your samples, especially in labeling, because sometimes these tiny mistakes in labels happen. I have seen that happen in classification studies. Think about the 3 R's. There's really a concept in experimental design: replication, randomization, reduce noise, reduce variation, by introducing all these factors in your models that filter out unwanted variation. And that's it for today, from my perspective. Yeah, from my perspective.
If you have any questions.
Sure, we can do questions. Yeah,
Yeah.
sure, that's a good idea.
So from 5 minutes for questions.
So,
Sure.
It worked quite well for, Univar again, because the what you say is you can have one.
Yeah, I addressed that in the conference.
Because the